We are going to go ahead and get started today. Thank you for being here. Good morning. It's an incredible Saturday morning, a great morning to learn about science, and a great morning for us to update you on the RAP study and explain to you how much of an impact this study has had on the entire field of Alzheimer's disease. My name's Sterling Johnson. I'm the principal investigator, the lead investigator for the RAP study. And I just want to welcome you and thank you for being here today with us. We're going to start this morning with our colleagues at the WAI Regional Milwaukee office who will welcome us and explain to us the impact of the Regional Milwaukee office. So I'd like to start by turning the podium over to Ms. Gina Green-Harris and Ms. Selena Ramsey. Welcome.
Well, good morning, everyone. Happy Saturday. Woohoo! I know you can tell that I am probably just a different kind of girl. So we're going to go ahead and get started. I want to welcome you all to the Milwaukee Info Session. It's honor to be here. I'm going to be joined by Selena Ramsey, who is our lead in Milwaukee with the RAF study. So we're going to do this together. And you see how she's excited about that, right? I'm just saying. Now, as much as I like this voice and have fun doing all these presentations, I just want you to know I am challenged with technology. You can't believe that because I'm only 25, but the reality is that if I don't click right, just okay. Don't just pretend like you didn't see that. So the info session outline is we're going to talk about there's going to be talk about new developments in the field. I believe there's going to be current status of the RAP updates, key findings, link studies, and what we'll do next. And then after that comes us. So I'm going to talk a little bit about the WAI Regional Milwaukee office, starting with who we are. Who is our team? And so we've got our team that you see all of our pictures. Many of you know these faces, so I won't go through them all, but I will acknowledge that Dr. Norris is not here. As many of you know, Nia, she's out today, so she is not here, but we definitely want to recognize her. I believe Ian, Selena, and Julie, and Heidi, and all the rest of the team are here today. But these are your Milwaukee folks that are supporting the RAP study that you see testing, you see phlebotomy, all of those visits. This is the wonderful team that makes that happen. Through our team in Milwaukee, we not only do the research, but we do outreach and education, in-home diagnostics. There's Stephanie Houston somewhere in the room. I don't have my glasses on, so I can't see her. But up there she is, Stephanie Houston, who does our in-home. Many of you may have met her if you have on your journey with your loved one. And she's also the director of the Amazing Grace Chorus that many of you know. We also do advocacy through our Community Advisory Board, and of course, research and philanthropy, where we know we need support to keep these programs going. Here are current research and outreach in our community. What you see is we have a vast and deep kind of connection with community. We believe research is really important, but research has to be connected to a give back, and it has to be connected to serving our community in general. So what you see here is all of the work we do in existence with our research program. It is really important that we meet people where they are. So although you're part of our research study, you also may need more education and understanding about the dementia journey maybe your loved one is on. We help with that. You may need some type of outlet for your own type of respite. We help with that. You might also need to understand diagnosing better. We help with that. So what we do in all of these areas is that we believe, even though research is critical to this work, serving the full dimension of humans and people is really the key part of this. Because at the end of the day, we can do the research, but without helping the end users, such as you all, we won't be anywhere. And we know we need to be of help and of service. So service is our priority to serve folks, to help in Milwaukee and beyond. So I won't go through all of these because it would take me a long time. And I believe that many of you can see. Can you all see that okay? All right. And you guys are like, please don't go through all that anyway. But those are I just wanted to clarify who we are and why we do what we do. And now we also have research, as I said. So I'm going to bring Selena up to talk about the RAP study as it serves you all in Milwaukee and the Milwaukee area. You ready, Selena?
Yes, ma'am.
Come on and talk about it.
Good morning, everyone. I hope you can hear me. I'm on the short side, so you might can't see me, but I'm gonna try to step to the side so y'all can see my face. So my name is Selena Ramsey. I'm the clinical research supervisor here at the Milwaukee office for RAP.
Talking to Mike. Talking.
Wait.
How about we?
Right, let me take it off. There we go. Like we do in church. Okay. Oh, there we go.
Well, now go over there.
Oh.
I'm gonna pull it for you.
Nope, this good. Okay.
Can they
So-
see you though?
Can y'all see me? Y'all okay? Good. So I just want to go over the support of the research team. So some of our community partners, I won't go through the list, but we partner with UW Reach for our RAP research program. We also partner with MCW where we run our RAP MRI visits. We also have a community advisory board where we get feedback for our research study. If there's things that we should implement or we should also put within the, like take out of the study. We try to get feedback from the people in the community, the people that we service on what we need for research. So when it comes to our research, it's not just things that we want to do, but hearing the voice of the people and implementing your guys' thoughts, ideas, and just also the feedback that you give us. And I'll go on to the next slide. There we go. So I want to go over some of the goals for recruitment and our program growth. So recently we have reinstated blood draws, MRIs, and also we're going to implement brief medical exams here at our Milwaukee office. A lot of you all have recently come in for a visit. So you have met our phlebotomist, Nicole, where she has done your blood draw for your RAT research visit. One implementation that we're looking forward to giving back to our participants is actually giving you guys back the lab results. Right now, we do draw labs for the research, but we aren't able to give you back those results. But we are looking forward to that implementation. So that can be a give back to our research participants and that all that you do for our research study. Also, we are doing MRIs at MCW. That's at the Milwaukee Medical College, or Medical College of Wisconsin, I'm sorry. And those are done at the Daniel Sorf Center. So you might see some participants with stickers on their name tags. Those are our biomarker ambassadors. So those are people who have either participated in either an MRI, lumbar puncture, or PET study. So even in this room, just talk amongst another after the session to get feedback on if you haven't participated in a biomarker study, asking other participants what was their experience like. And it's just about building a community, knowing that we all have each other just for education, talking to each other about each other's experiences, and being part of this research and this overall cause to hopefully one day end Alzheimer's disease that affects all of our families in some way and capacity. And then also just to go over the implementation of the brief medical exams. Recently, we have hired a nurse practitioner. So we look forward to her coming on board and being able to offer participants an interaction with a physician at their actual RAP appointment. So currently we don't have a nurse practitioner, but now as the NP becomes on board, you can ask them general health questions about your health. If you have concerns, you have a physician at your actual visit to talk to. So those are some new things coming up in Milwaukee, and we're very excited about to give back to our participants because you all give your time, just contributing your time to the study, but also just your vulnerability on how the disease affects your family, because that's one thing I believe that we do all have in common. Next is the recruitment goal for RAP. So this year, we're working in our 5-year grant. One of our goals for the grant is to enroll 200 additional African Americans. So this year we implemented our campaign where we asked the community, be one of 200. In this campaign, we ask for your help to help us enroll additional 200 African Americans in this study. We asked, and you guys have answered. Within this time, we have had so many new interests within the study, even getting friends, family, all enrolled just based off the cause of wanting to find a cure or either preventing the onset of this disease or investing in interventions to help learn more about it. And with this campaign, the goal is for 5 years. Right now, we're about 2 going on 3 years of our goal of this grant goal, but we have already went over. It's about 70% of new enrollment. We're reaching the 200 goal of new participants. So that wouldn't happen if it wasn't for the people in this room spreading the word of RAP, talking about your own visits, and just how the study has helped you all. So I just want to take time to say thank you. Thank you for your contributions, for your vulnerability coming in, because it means a lot to the study and just for the cause itself. All right. Oh, and that's okay.
All right. Thank you so much, Selena. And Selena was fantastic, wasn't she? Give her a round of applause. She was a little nervous. But I just want to say, we want to thank you guys so much for your RAP participation. We've got folks who've been in the study for 20 years, all the way to folks who have just joined this first year. 20 years, or how many of you guys have been here several years, or at least, okay, let me see my 1 to 5 year folk. All right. How about my 5 to 10 year folks? Give yourselves a round. How about my 10 to 15 year people? And then my 20 and over, my 15 to 20. Whoa. And anybody who was beyond that, I don't think there was to be on there, but if you remember it, I don't know. I'm just saying. But we want to thank all of you for all that your contributions have made. And I just want to do this really quickly, and I know Dr. Johnson Sterling will probably talk about this, and perhaps Nate will as well. But I want you to know, those of you who are wondering why are we seeking African Americans to enroll in the study, is that it's really important that as you are noticing, there are lots of interventions, there are lots of new ideas, and a lot of new ways in which we're addressing Alzheimer's and dementia. It is so important and critical that we have everybody included in that research so that we can make sure that whatever intervention is coming to fruition, that everyone can benefit from it. But also, it makes better science. It just makes better science to have everyone included in the study. Does that make sense? And so I just wanted to acknowledge that we're not forgetting a general population or everybody. We're hoping that everybody at one point will be involved and engaged in this research study, because guess what? Alzheimer's, dementia, and other related dementias and whatever, Lewy body, none of that cares about race and ethnicity. None of that cares about any of that. So we have to think of it from a human perspective and making sure that we get the answers for everybody. Does that make sense to everybody? And with that, I'll invite Dr. Nate Chen to the stage to come and talk about why RAP. Thank you so much for your time. And we will have Q&A after.
Thank you, Gina. Thank you, Selena. All right. Thank you all for being here. Can you guys hear me okay? No. All right. All right. Let's see. You know what? I'll just hold it. That's no problem. Okay. Well, thank you for that introduction. Thank you all for coming today. We're always excited to give our RAP information sessions. We like to share the things that you guys have created, that you have contributed to. And if we don't say it more than 10 times today, thank you. But also, the work that is being done here really is leading the field. And I know that might sound pretty typical of a scientist to come up here and say, oh, we're leaders, but we truly are seen as leaders in this field. And the data that you all create is what really has changed how we view Alzheimer's disease and the different causes of dementia. And so what we're seeing now, and what we're trying to really do, is separate biology from the things that we experience. And so in this slide here, we start with this biological definition. Alzheimer's is not dementia. Alzheimer's is this buildup of proteins that really shouldn't be in the brain. And it's amyloid and it's tau. And I'll go into that in just a minute. But in addition to that, though, what we care about, of course, is not not just biology, it's the things that we experience in life. And so the clinical stages are going to be defined separately. And that you're going to hear me say mild cognitive impairment, dementia, the various stages of dementia. But what we're learning as we go through this, as we disentangle Alzheimer's and the symptoms, is there's actually this really long asymptomatic or pre-symptomatic phase of these brain pathologies, Alzheimer's in particular. And in fact, someone could have the changes of Alzheimer's 10, 20, 30 years before they're diagnosed with an impairment in clinic. And that really came about because of you all and because of what RAP is able to do and show through longitudinal visits, through people coming in year after year, giving their bodies and their time to the scientists. So we now can detect these changes. It started with spinal fluid and then PET, and RAP was actually known for being a leader in the PET space, but now it's blood. And that is really revolutionary. And there were many in the field who didn't think it could be done, and it is done. It was FDA approved just earlier this year. And that was because up to 40% of the data the FDA used came from Wisconsin and from you all here. Once we can identify a protein, well, then the next step is, well, how do we stop it? How do we remove it? How do we prevent it? And that's really the space that we're in. And there are therapies now that we are using in clinic to remove the protein. And I'll talk about that in just a second too. But there's one thing, and Gina mentioned it, that's really important, is that we don't live in isolation. Our brains are not isolated from one other disease process. It's not that we have pure Alzheimer's. There are lots of other things that affect us. Some of them are brain diseases like Lewy body and vascular and Parkinson's. And some of the other things are life experiences, as well as other medical conditions. And they all can influence the symptoms that one experiences in mild cognitive impairment and dementia. So they all matter, and we need to look at those as they relate to these proteins, but also just as they relate to the lived experience. And so when you come in and you give all of that information, and I know it's a lot that we ask of you, but it is really valuable for us to understand not just your biology and your age and your genotypes and your genetics, but really the things that you experience in life and your health factors and your medications, because we know those things matter as well. So I just wanted to emphasize dementia is a different thing than Alzheimer's disease. Alzheimer's is biology, the two proteins, and dementia is really this collection of symptoms and things that we look for in the clinical space. I like to say to my patients, really, mild cognitive impairment and dementia, it's our way of trying the best we can to understand the things you're experiencing. It's the lived experience of having a thinking change, whereas Alzheimer's disease is that biological process happening in the brain. So dementia is not normal aging. It's not Alzheimer's disease. It really is just a way of staging a person's thinking change. It is the consequence of disease. It is the things happening in the brain that eventually cause the symptoms of dementia. The 5 or 6 main brain diseases you can see here, Alzheimer's is the most common, is amyloid and tau, but we also see a lot of vascular disease. When you get those MRI scans, it's really helpful for us to understand blood vessel changes in the brain. It's very common, along with, as we're starting to learn more, Lewy body changes and Parkinson's changes. I said Alzheimer's is amyloid and tau. Lewy body and Parkinson's, it's a different protein or 2 proteins, alpha-synuclein and Lewy bodies. And so it's completely different from Alzheimer's, yet they occur quite frequently together. And so why is that? And so we need to understand that. Frontal temporal disease is a different protein, and it actually relates to this other one called LATE. LATE is an acronym that stands for limbic predominant age-related TDP-43 encephalopathy. It's a horrible name, right? Horrible. But the key part of that name is TDP. And so that's just a different protein that builds up in the brain. It actually looks a lot like Alzheimer's disease. And so right now we don't have the ability to detect this protein the way we can in Alzheimer's with amyloid. But our program in Wisconsin is working on that. We believe we'll be at the front of being able to develop those tests. So ultimately, the things that we experience are because these proteins or vascular changes exist, and they influence our brain cells, the health of our brain cells, eventually leading to early death of those brain cells. And so when I think about the process of Alzheimer's disease, it does start with amyloid. And that doesn't mean we think that Alzheimer's is only amyloid. We know there are other important factors. But when that protein develops, that sets off this cascade, this trigger of other processes in the brain that we now need to identify, understand, and intervene on. And so when a person develops elevated amyloid, some time goes by, it could be anywhere from 7 to 10 years, and some individuals are going to start developing the tau protein, the second protein of Alzheimer's disease in the brain. So amyloid and tau together. And then with tau in particular, there is that disruption in the health of the brain cell. The brain cells are going to pass away, they're going to die, and they're going to have neurodegeneration, atrophy of the brain. All three of these things can happen without a person ever having symptoms. We learned that because of RAP and RAP participants. Before we did, it was really hard for us to differentiate what a person was actually having in their brain. Eventually, there's enough brain changes, and they're going to go into a clinic, and they're going to get diagnosed with mild cognitive impairment. They're going to experience a thinking change, have some testing that shows those changes. And then because we can't stop the progression of these proteins right now, they will progress, and they will develop then dementia. We are doing an incredible job at identifying vascular risk factors, treating blood pressure, cholesterol, diabetes, our weight, our sleep, other factors. But even with that, we still are seeing increased rates of dementia and Alzheimer's disease. And so why is that? Well, one of the consequences is people are just living longer, and age is the greatest risk factor for developing these proteins and developing cognitive change. And so in this figure here that comes from the Alzheimer's Association, where we see an increase from 6.1 million people in 2020 to 13.8 million in 2060, it's because people are older. This older group of individuals, 85 plus, are going to be developing these changes. And so that's the baby boomers. But what we do see here is overall stability in our younger 65 to 74 and 75 to 84. People are still developing these things, but we're actually having an impact on the rates of change. So in Wisconsin, we see this year 130,000 individuals who are 65 and older that have Alzheimer's or a related brain disease causing their dementia. 130,000 sounds small, but it's not, and there's even more caregivers. So they say here 191,000 caregivers of people with dementia in Wisconsin. I suspect it's much higher than that, but we just aren't asking as many people. It's the 7th leading cause of death in Wisconsin, and it is a very costly condition, not just for healthcare, but for families as well. I brought up this idea of mixed disease or having multiple different things happening in the brain. It's a very active area of research. So in addition to identifying these proteins for Alzheimer's, we need to identify what else is going on in the brain. And 2 of these studies here, the one on the left is recent, 2024, and the one on the right is from 2022. But what they did is they used donated brains. So people who were research participants that passed, that donated their brain, they looked at those to see what exactly is happening there. These new tests, blood, spinal fluid, PET scans, are great, but it's not definitive the way we think of autopsies. And so what we're seeing is that 1/3 of these cases are going to be pure Alzheimer's, and 2/3 roughly are going to be mixed Alzheimer's and something else. This has changed how we view treatments. Some of these are going to be old, since 1996. Treatments that we use right now in clinic for symptoms, but then the purplish ones are the new ones. These are new drugs to remove the protein of Alzheimer's disease. Lecanemab and donanemab, and one is actually in the middle to help with symptoms of behavioral change. This is the only scientific slide I think I'm going to share with you today. And so what I'm going to show you here is that these drugs are effective at removing amyloid. So in this picture here, these individuals, this is group level, so this would be a group of participants, they had 80 units of amyloid removed with this drug in the phase 3 trial. So that's a pretty significant amount. Someone with Alzheimer's dementia would probably have about 100 units. But then this is what we see here clinically. People on the drug are still declining, but they're declining at a slower rate than those not on the drug. Meaning after a year and a half, they have more cognitive function, more physical function compared to those on the placebo. And so one could think of this as a preservation of about 3 months per year that they're on this drug. And so with that in mind, that's the context we wanted to share with you. I'm going to bring Dr. Johnson up, and he's going to talk about the RAP study specifically.
Thank you, Dr. Chin. Thank you, Dr. Chin, for that. That was an incredible overview. And you see already the impact RAP is having on our field. We're understanding the biology better than ever because we can understand the longitudinal progression of this disease. I'm just going to take a moment to zoom in to RAP now that we've seen kind of the broader impact on the field and where the field is at. RAP is both a longitudinal study in and of itself. We call it an observational study because we're not doing drug trials in RAP right now, but we want to talk to you about that. And in addition to being a longitudinal observational study, it's also a registry for a bunch of linked studies. And you'll hear from some of our linked studies today. We'll talk to you about that. Well, who is RAP? These red dots are where you all of our RAP participants live. It's a pretty incredible to see that all over Wisconsin and spreading throughout the country even. 1,938 people in the study. There's 1,142 of our RAP participants that have had a visit in the last 4 years, so that's kind of our active pool, you might think, we might say. 283 new participants since 2020. And we've got a new visit location in South Madison at the South Madison Partnership Office, which is helping us be more convenient to the community. We already heard about the 1 of 2,000 or 1 of 200 goal from Ms. Ramsey, and it's really gratifying to see that RAP is now 26.7% from diverse groups, particularly African Americans, and we're just excited about the partnership with Ms. Gina Green Harris and Selena and the team that have made this a success. Our participants overall have been in the study 14 years on average. 69% have a parent with Alzheimer's disease. 70% are women. 39.6% of our participants have the ApoE4 risk gene for Alzheimer's disease. And the average age is 66.7, 67 almost. But you see there's a big range in the current ages of our participants, and that's what that bar plot is there on the left side. Well, what about our health conditions? 67% of the RAP participants either have high blood pressure or are being treated for it. And 63% have high cholesterol or are being treated for that. 15% with diabetes, and 38% with a high BMI, body mass index. So that's some of the health characteristics of the study. What about cognitive impairment? Most of our participants, most of you are unimpaired. That's what CU means, cognitively unimpaired and stable. CUD, they're that 18.2% cognitively unimpaired, but we think there may be some decline that we're noticing. Only 6.3 have reached this mild cognitive impairment stage, and 2% the dementia stage. So we're very much a pre-symptomatic or asymptomatic cohort still. These are the things that you all do, and you probably know these better than I do. The imaging that we ask you to do, thank you for doing that. The MRI scans, the amyloid scans, the tau scans. We ask for lumbar punctures. We're now going to be asking for carotid ultrasound, and we'll be doing that here in Milwaukee very soon. We do, of course, the memory tests, the questionnaires, the endless pages of questionnaires. Thank you for doing those things. The medical exams, and new to RAP is this smell test. It's called the UPSIT, the University of Pennsylvania something test, smell test. It's olfaction is actually a symptom of Lewy body disease, having a deficit in olfaction or smelling ability. might be an indicator of dementia. There's other reasons for us to have olfaction deficits, but that's one of them. Okay, getting back to some of the numbers of who we are in RAP, 740 of you have done at least one amyloid PET scan. Raise your hand if you've done an amyloid PET scan, if you've been in that PET scan. Thank you. 702 have done a tau PET scan. That's measures these tangles that are more associated with symptoms. 8,500 blood samples from our RAP participants. 954 people have done at least one MRI. Raise your hand if you've done an MRI with the RAP study. Incredible. And nearly 600 people have signed up for brain donation. And we're not in any hurry for brain donation, but we hope you'll consider that. Raise your hand if you've signed up for brain donation. Amazing. Thank you. Brain donation is important because we can look at the brain under the microscope and see what is actually there, what pathologies are present. Are there Lewy bodies present? Is there this TDP protein present? Are there vascular changes present? It helps us have a gold standard that's going to help us develop new biomarkers for other diseases in the future. Okay, well, what discoveries have we made? We've been, I need my glasses on for this. I'm going to look at the screen. We're going to talk about clocks, about the temporal information we can get from these biomarkers you're doing, from the PET scans and the blood. We're going to talk about the impact of non-AD proteinopathies or pathologies and how those might influence progression rates. We're going to talk about the ability to detect these other diseases, and also talk a little bit more about blood in the next few slides. And so you're already hearing all this other stuff besides Alzheimer's that we're talking about. It's because of studies like RAP and just advances in the field that we're able to now say there's these other diseases that are present, they're contributing to symptoms, and they probably have a long progression rate just like Alzheimer's disease does. Well, thinking about the PET scans first and the biomarkers, I know we're all worried about do I have Alzheimer's disease growing in my brain? Is this going to happen to me? Here's some numbers to think about. Even though 70% of the people in the study have a parent with Alzheimer's, only 29% of us, 29% of RAP are amyloid positive. So that's an important thing. 21% are tau positive, meaning there's evidence of tau protein in the brain. When we look at the blood, we're just looking at it the other way now, 61% of you are very much negative for Alzheimer's disease proteins in the blood. That means we're not detecting it in your blood. There's about 20% that are in an indeterminate intermediate zone, and about 20% that are in the definitive clear zone for Alzheimer's disease. So that's where we're at with these blood tests. They very much agree with the PET scans, and they're really going to help not just this kind of study, but help the entire field be more scalable. Physicians will be able to use blood tests instead of very expensive PET scans in the future. RAP is driving new research. One of the things we've done is launch a study that is called Clarity, that is largely based on the findings from RAP. And it's being launched all over the country, taking what we're learning from you and taking it nationwide. We are raising awareness for this multi-etiology idea. We've recruited new faculty to the university, including the person who invented tau PET. He's now a faculty at the University of Wisconsin. His name's Hartmut Kolb. And yes, we like that. And also Professor Henrik Zetterberg, we've recruited him to the University of Wisconsin. He's going to help us develop new biomarkers for these other diseases that we've started talking about, TDP, Lewy body disease, and so forth. And we're continuing to develop partnerships with industry and any other stakeholders that can help us get to answers faster. And speaking of faster, I got to move faster here. Current research questions. Are discoveries generalizable to all communities? That's something that's very important to us, as Gina mentioned. How do lifestyle and medical health factors impact cognitive change? How early can we detect AD as well as these non-AD proteinopathies as they're beginning to emerge? How can we expand the time between the detectable stage of the disease and eventual symptoms? That's really what we want to try to do, is give people healthier lives for longer. How can RAP inform clinical decision making? And we think all of these things are very feasible with us. Well, just a few quick highlights of some of the science that we've been able to do. One is this idea of a biomarker clock. And what do we mean by a clock? Is that with these PET scans and with the blood tests, we can use those things to estimate when a person, what age they were at when they became abnormal on that blood test. And therefore, we know at any given point in time in their future how long they've had disease in their brain. And we call these biomarker clocks because they're going to help us really understand how things progress on a person level basis. And this is important. You can kind of see it here that each of these people in red, they're amyloid positive based on their amyloid PET scan. You can see that they're all kind of increasing at the same rate. The reason there's heterogeneity in these so-called spaghetti plots is because they're at different ages at which they became amyloid positive, but their change rate is going up about the same rate. So we can fit a simple mathematical function to that, and we can come up with this idea of an amyloid clock. This is what it means in reality, is that for each of us, Alzheimer's is going to present differently. The age at which a person becomes amyloid positive is shown here. There's a span from 40 to 80 plus, and it would go longer probably, but that's just the age sampling restriction of our study is at 85. So there's a whole 40 to 50 year span of time at which a person could become amyloid positive. We have been observing this, but our colleagues up the road at Mayo Clinic, at down the road at Wash U, down in St. Louis, in Baltimore, and the multi-site ADNI study, we're seeing this very same thing, that amyloid doesn't care whether you're a marathon runner or a couch potato, it's going to grow at the same rate. What we do think matters, and we do think exercise and healthy lifestyle is important, is because that's going to slow down the cognitive change. But it doesn't seem to be impacting these longitudinal rates of amyloid. We're able to do a similar concept for the tangles. Alzheimer's is plaques and tangles. On the tangle side, we're able to also fit this to a temporal framework to understand the age at which it began and how long it's been in the brain. That's going to help us understand things like risk and resilience in a whole different way than we could before. The usual way of studying risk and resilience is to wait till someone ages to age 85 and declares themselves still cognitively healthy. Now we can anchor that to the age at which they began their amyloid journey, and we can see how many years they've been symptom-free relative to their very own age at which their journey began. We can do this for vascular disease as well. This is why MRI is so important. Thank you all for doing that. We see these little white spots on that MRI scan. These are the areas where there's a low blood flow in the brain, and we can quantify that, and those are these dots here on the plots. And we see that these things kind of grow at the same rate as well, meaning we can do a vascular disease onset age and disease duration at the individual level. These aren't group effects anymore. They are individual level effects, which is really going to be helpful in the clinic. Plasma, this marker called plasma p-tau217, which is now FDA approved thanks to the work of RAP and many other studies that work together to make this happen. We can also fit that to this temporal clock idea. Well, how can we use this information? What we have here in this scatter plot, we call these spaghetti plots because each person is a line, and we get to see where these lines go and don't restrict these lines. These are observational data. So on the x-axis, we have their amyloid time or amyloid chronicity, how chronic that disease has been. And on that y-axis, the vertical axis, we have a person's symptoms. And we see that on average, which is that bluish line, that it takes about 20 years in our study to reach symptoms, 20 years of being amyloid positive. But there's many people who are ahead of the curve who are reaching symptoms before their amyloid load would predict. Are now seeing that these individuals likely have vascular disease in their brain. They may have more likelihood of having alpha-synuclein or Lewy body pathology in their brain. So this is helping us expand our understanding of a person's individual journey. RAP has moved really quickly into the blood biomarker space, and this is because of those new faculty that I mentioned, especially Dr. Zetterberg has led the way in this entire field, and he's really presenting RAP with lots of new opportunities, and we're making the best of those. Well, we are, that's kind of a quick whirlwind about where we are now in the study. We're going to open it up for questions in a bit, but we also want you to hear about the impact that you're having in all of our linked studies. So let me now turn this over to Dr. Nate to talk about microbiome.
All right, thank you, Sterling. And we're going to have some of our great researchers come up, and so if you guys are ready, please start making your way down to the front. This one is from Dr. Barb Benlin, amazing researcher in our team, really looking at stool or poop. And she's starting to show that it does matter, that what we take in is important. And what we're seeing is that those with Alzheimer's disease have a different microbiome in their gut. And so this is all through participation in people donating stool. There's less diversity in people's stool when they have dementia and Alzheimer's disease. And there's certain organisms that she's starting to find are more prevalent, and they're the ones we wouldn't want. And so she's starting to look at why that might be. And one of the things, and maybe you've heard of this term, leaky gut, this idea that our stomach is inflamed, it leads to separation in some of the cells, and there's inflammation that happens. It goes from the bloodstream even into our brain. And so she is looking at that, and she really is at the forefront of this. I don't think she would say it, but I think she really is the leader in this field. There is a study that she is conducting right now, also looking at, also looking at family members of those who have Alzheimer's disease. And if you're interested in donating your stool to us, please, there might be a table. Yep, there is a table over here. It can all be done from home. You don't have to do it with us. So it's in your privacy of your own bathroom. But it's a great study.
Thanks, Nate. We're going to hear from another one of our studies here in just a second, but I want to let you know that we share RAP data throughout the world. It's anonymized or de-identified so that it's not, they can't identify you, of course, and that's part of the consent. But we've shared our data with other investigators from Harvard, from in Australia, in other parts of the world, and we're making faster progress in our field because we work together with other scientists and investigators. Now I want to turn it over to Ms. Leah Sanson, who's going to talk about one of our other studies. Whoops, sorry.
Thank you so much, Sterling, and thank you, Nate, for the hand up the steps. Hello, everybody. Thank you so much for being here today. We appreciate it so much. We wouldn't be here without you. I just wanted to start out with a thanks for everything. So I'm here speaking on Dr. Kimberly Mueller's behalf. And so Dr. Kimberly Mueller has been studying speech, language, memory, and other aspects of cognition in RAP for many, many years now. And so you might think, why is studying speech important? So speaking is something we do every day. Speaking uses many brain regions and functions, and changes in speech can actually reflect how the brain is working. And so our goal is to find how it's maybe easier at-home ways to track brain health. And many of you have allowed us to record your speech in testing over approximately the last 10 years. So thank you so much for that contribution up to this point. We have published many findings with the RAP speech data that you've provided to us. And so we've shown that changes in speech track with changes in memory and thinking from other cognitive tests that you do with the RAP study. Another thing is that speech pattern changes are also linked to AD brain biomarkers, which is very important in tracking. And finally, we have shown that some modifiable risk factors are also linked with changes in speech, such as social engagement, depression, and head injuries. And we think this information will help us develop non-drug cognitive therapies, so for people at risk of dementia. And we're excited to continue working with your speech recordings in general. RAP is known as having the largest longitudinal data set of speech recordings, and that's all thanks to you. So thank you so much. And we are also currently recruiting for a study related to speech and Alzheimer's disease changes. So check that out on your way out. Thank you so much, everybody.
Thank you, Leah. RAP is having an incredible impact because we've got faculty partners who just make the study better by augmenting. One of the things we mentioned was understanding risk and resilience, and we're fortunate to have an amazing team who's taking on the study of resilience, and I'm going to turn it over to Ira to tell us about that.
Thank you very much. Good morning. My name is Ira Frommond. I'm one of the scientists in the Okonkwo lab. And in our lab, we like to research modifiable and non-modifiable risk factors that are related to cognitive resilience with aging and Alzheimer's disease. And an example of a modifiable risk factor would be your fitness level. Maybe you're not very fit, you can do something about it to become fitter. Or if you're currently fit, and you ignore your fitness, you can become less fit, right? And then non-modifiable risk factor would be our genes. There's not much we can do about those. We are pretty much stuck with what we got, right? And here is a pretty current photo of our lab. You might see some familiar faces there if you've participated in one of our studies. Our fearless leader, Dr. Ozioma Okonkwo, was here today. Can you wave? Thank you. Great. And the list of our active studies currently is a study called LIFE. It studies the relationship between cardiorespiratory fitness and then biomarkers of Alzheimer's disease, such as beta amyloid and tau, and also cognitive features, right? And if you participated in that, Alyssa, the study coordinator, is here today as well. And our current study, CLOTHO, is looking at a non-modifiable risk factors, so our genes, and how that relates to synaptic function and cognitive performance. So synapses are little spaces between neurons, the cells in our brain, that allow them to talk to one another, and those change as we age. And study coordinator Vinao is here today as well. And I will today highlight some of the findings from these two studies, but we also have currently active super aging study, and Monica, study coordinator, is here with us today. And that is a study that looks at exceptional cognitive resilience with aging, and Replenish study that looks at the relationships between sleep, plaques, tangles, and cognitive performance. All right. So first, we'll go with this. First, CLOTHO study. So non-modifiable risk factor, what we know is that people who carry a certain variant of this gene have higher concentrations of CLOTHO protein circulating in their system, in their body. And this higher concentration of CLOTHO is related to longer lifespan, less risk of age-related diseases, and better cognitive performance. And what we know from our own work is that they also have better memory, better executive function, slower rate of cognitive decline with age, less amyloid, even if they carry a risk gene for Alzheimer's disease, less tau and slower rate of accumulation of tau. And if you've participated in this study, we've asked you to undergo lumbar puncture, obtain small amounts of cerebrospinal fluid, and gotten levels of beta amyloid and tau from your cerebrospinal fluid. We also know that people who carry the certain variant of this gene have less neuroinflammation, neurodegeneration, and better synaptic function or less synaptic dysfunction. And those we can get also from your cerebrospinal fluid and blood. And for synaptic function, you're also undergoing PET scans. So we're getting levels of your synaptic function or dysfunction, however you want to look at it, from blood, CSF, cerebrospinal fluid, and PET scans. And with, I think the one slide is gone, but overall it said, if you participated in our life study, that people who have better cardiorespiratory fitness seem to experience less cognitive decline with age, even if they're accumulating beta amyloid and tau, and it also seems protective against vascular changes, as Dr. Chin said, while not main features of Alzheimer's disease, they are contributing to the development and progression of the disease. So with that, I would like to thank you for your time and dedication and participation in our study. And because of you, I was able to share some of these findings with you today. So thank you again. And with that, go Brewers.
Thank you, Ira. I'd really like it if you guys could somehow create a way to create more clotho for the rest of us, though, even if we don't have whatever mutation you're talking about. All right, let's see here. Did I go backwards? There we go. It's Dr. Zulsdorf here.
Thank you, Dr. Chin. So it's so nice to see all of you today. I am Megan Zielsdorf. I am a scientist. I am a public health researcher in our UW-Madison School of Nursing who has been studying modifiable risk and protective factors like stress and social connectedness in RAP for, I realized today, over 15 years. So well, what I was going to say is it's really nice to be in a room with some of you that have actually been with RAP even longer than I have, because there are only a few of us. I'm actually here today to represent Dr. Carrie Gleason and talk about the study, African Americans Fighting Alzheimer's in Midlife, that she directs in partnership with Ms. Green-Harris and the Milwaukee team here. This really speaks to one of the first points that Dr. Johnson talked about today, where we are focusing on doing science that is generalizable, that applies, that provides benefits for all populations. This is really critical to the Wisconsin idea, right, that RAP embodies so well. And AA-FAME, which is what this study, the shorthand we use, is part of that. This is making sure that Wisconsin's Black and African American communities are included by and benefit from the Alzheimer's science that the UW is doing. We do that through, I'm glad I don't have to explain much of this slide because Gina did a wonderful job of talking about the foundational work of community-based participatory research early on. This is just at its heart the idea that Madison doesn't just partner with other scientists at Milwaukee, but we actually partner with our participants and the communities around them that as experts, where they let us know how we can do better science, how we can make sure that our research questions address things that are important to them, that our research studies are relatively easy to participate in, whether you're just enrolling today or you enrolled 20 years ago and you want to keep going, right? That the participation is as easy and as enjoyable as possible, except for the part where you have to learn all of the series of numbers and then repeat them backward, right? We've given up on that. That's never going to be enjoyable. But we try to make the experience a good one for you. We try to make it, participating in research, feel like a good and natural idea. I'm just going to really quick cover a couple of really exciting papers that have come out of AA fame in the past year. The first one was a paper that Dr. Gilda Ennis wrote up, a study that she did looking at our AA fame participants. Again, this is really focused on making sure that these exciting new blood-based tests where we're looking for preclinical Alzheimer's disease, that they work just as well across all populations. This is a really key question to ask. What Dr. Ennis looked at specifically was whether the presence of chronic diseases that we know are really prevalent in Wisconsin communities and actually in the US population as a whole, like diabetes, like cardiovascular disease, whether these might influence how well the blood tests work. And the good news is that in our AA fame participants, our Black and African American participants, I will say it worked pretty well. One thing though that we found that was really important, and it's important for both African American communities and far beyond that, is that we do need to pay attention to kidney function in our patients or anyone that we're screening, because it does look like impaired kidney function can actually elevate the levels of p-tau217, that marker of brain amyloid. It can elevate the levels in the blood a little bit beyond what levels of amyloid are in the brain. So clinicians who are conducting these tests of screening just need to be aware of any kidney disease or any impaired function in their patients. But overall, this is really promising findings. Whoops. The second study I want to talk about, Dr. Diane Gooding really discussed this community-based approach that is based on what Ms. Green-Harris was talking about earlier in another publication. So some of you might know that a couple of years ago, Gina Green-Harris, Nia Norris, Stephanie Houston, they actually put out a paper that described all of the work that they were doing with all of you, that the regional Milwaukee office was doing with all of the participants it was serving. At this point, over 50 teams across the country have cited that paper in their work. They've been inspired by the approach. We took that approach and we adapted it for the Madison Black community. And Dr. Diane Gooding actually now has really dove deep, described that, and put it out into the world. And this is how we bring not just your data points, your cognitive tests, your blood pressures, but actually your voices and your perspectives out into the scientific community so that other teams can hear what you have said, the information you've given us. With that, I'm going to turn it over. Thanks so much for having me.
Thank you, Megan. The last thing we wanted to share is from our colleague, Dr. Lindsay Clark, who is really leading our Wisconsin effort in returning these results, research results, back to participants. We have learned through the things Megan and Gina have already talked about, the importance of communication, transparency, and giving back. And so Dr. Clark is studying what that looks like in the space of returning biomarkers. And really, it's because of leaders like Dr. Johnson that have allowed for us this space to be able to interact with you great people about sharing what are we finding? What are these actual results meaning? How do you interpret them? How do you feel the process is going? And so far, things have gone quite well. If they haven't, please talk to me after this presentation. I would like to hear more about that. But what we've done is really share amyloid PET scan results with our participants. But we want to do more. We just have to do it in this structured, methodical way and prove to the field that this can be done reasonably and that people want this information. And so Dr. Clark has really been at the forefront of showing that. These are some publications to really highlight that, but it's all because of your willingness to not only do the PET scan, but then to go through this process with us in learning what this is, what it means to you, and what you do with that information afterwards. And what we're seeing, actually, I just want to draw your attention down here. What we see is that people find this information to be influential. It does impact how they think about their future, their lives, their willingness to engage in some of the lifestyle changes that Ira talked about, as well as now we believe clinical trial interests too. And so with that in mind, I'm going to turn it over to Sterling to wrap us up.
Great. Thank you, Dr. Nate. And thanks to all of our linked studies. What is next for the RAP study? Given the state of the field, what we know now from you, what we know now from reading the literature and interacting and collaborating with colleagues across the world, what do we want to do next? As Dr. Chin mentioned, we're going to be disclosing more results. We want to empower you with the information you're providing us and make sure you have that. Whatever's interpretable and clinically actionable, we want you to have that. So we are working on a pathway to do that. We're also, RAP is really expanding. It's more of an international study now, and it's taking a bigger leadership team. Dr. Chin is part of our leadership team. He's been our medical director for years, but he's now part of the MPI, the multi-PI team. That's what we call the PI structure. And also Jessica Caldwell has been recruited to the University of Wisconsin. She has particular expertise in prevention, and she will be part of our leadership team as well with the RAP study. We're excited about that. And where are we going to take this study in this era where there's prevention research happening, there's clinical trials on removing amyloid plaques in people who don't have symptoms? If these things turn out to be useful and positive, we want to be ready for that. So we are paying very close attention to prevention research studies, clinical trials, giving these amyloid therapies to people who don't have symptoms. And we're considering whether we should do that in the RAP study itself. And so we would like for you to look at the survey that's in your packet. There's a survey about what's your temperature about an intervention. If you knew you had these amyloid plaques in your brain, and knowing what you know about how these things progress like an escalator, would you want to have those amyloid plaques removed if we could do it in a safe way? There's a survey there, and we'd be happy to have that be part of our discussion in just a second here. And we also just want to continue what you're doing. Stay with the study as long as you can. The longer you're in it, the more valuable your data points are to the entire world. And so we thank you for being a volunteer. We hope you'll consider lumbar puncture so we can get spinal fluid, because that's where these other proteins like TDP-43 and alpha-synuclein, that's where we can detect this stuff, and that's how we'll get to a blood test. Like we did for Alzheimer's, we want to do the same thing for TDP-43 and alpha-synuclein, because those things co-occur with Alzheimer's and confuse the diagnosis. We also want you to just continue holding steady with the study. Come in every 2 years or so for your visits, do the blood draws, do the imaging visits as much as you can, volunteer for these link studies as much as you can. We appreciate you doing that. And finally, we just want to thank you from all of our staff. Would all of our staff, if you'd raise your hand, any of our staff here? Thank you. Thank you to the staff for making all of this possible. This event today, as well as this study and the whole program of studies that are linked to RAP. So thank you to our staff and all of our staff. We all want to thank you, our participants, for being a part of this study. And with that, let's open it up to your questions. We want to hear from you.